Then there we go, that's what I was waiting for. Then we're going to get into the speaker presentation, A Day in the Life of a Sales Guy, Adventures in Pharmaceutical Development by Tobi Williams, followed by a Q&A and a brief poll. So a little bit about the ACS for those that don't know. It is the world's largest scientific community where members have access to career development and networking, member groups and communities, conferences and events, as well as funding and awards. There we go. For career development, ACS offers the CNE and Jobs Database, Career Pathways Workshops, ChemIDP, the Individual Development Plan, and free career counseling. The career consultants hold free virtual office hours for ACS members on the first Thursday of every month from 12 to 1:30 p.m. If you're interested in that, you can look up ACS Career Consultant office hours, and I believe it'll ask you to log in, and then you can sign up for those. And there we go. It looks like the next one will be on July 6th. The topic will be, is grad school right for me? The ACS member groups and communities are inclusive groups and communities for all. So we have, just to name a few, there are so many more than this. The Women's Chemist Committee, Minority Affairs Committee, Younger Chemist Committee like us, and Chem Clubs. The ACS conferences and events are held in a huge range. There's the ACS national meetings, which are held twice a year, ACS regional meetings, which are held, I believe, once a year by region, and the ACS Green Chemistry conferences. The upcoming national meeting will be in San Francisco, and the topic will be harnessing the power of data. And the upcoming regional meeting is going to be CERMAX, which is going to be ChemCon, the chemistry convention, in Durham, North Carolina on October 25th to 28th. There we go. A little bit more about the YCC. We are younger leaders transforming the world through chemistry, which means that we advocate for, develop, and support rising chemists like you to positively impact their careers, the ACS, and the future of chemistry. If you're interested in getting involved, you can attend YCC events at ACS national meetings, or come to events like this one. You can join or start your own local section YCC if you'd like to reach out to us at the email that was being used to send the Zoom link to. You're more than welcome to. You can also find us on social media, or you can apply for YCC sponsored awards. As you can see, we do a ton of fun events. We do professional development. We've done career panels for undergrads and grad students. We've done brewery and cidery tours, as well as lab tours. There we go. If you'd like to find us on social media, you can do so by looking up ACS VA YCC, ACS Virginia YCC, or Virginia Younger Chemists Committee. Any of those will pop something up for us. If you'd like to scan the QR code, I will give you a second to do so now. And we have also recently started a YouTube channel where our recorded talks and presentations like this one are uploaded. Finally, the Virginia YCC has been nominated for several Chem Luminary Awards. In 2021, we were awarded the Best Activity or Program Stimulating Member Involvement. And in 2022, we were awarded the Outstanding Continuing Public Relations or Communications Program of a Local Section Award. Hopefully more to come. If you have any questions during the presentation, you can ask them in the chat, or I believe Tobi is going to have his own set of rules where he will ask you to unmute and just jump in. Or after the presentation, there will be an open Q&A session if you'd prefer to wait until after. And I'd like to take a second to introduce our speaker after I get my video out of the way. There we go. So Tobi Williams is a native to Virginia. He has worked in several companies in the area, including Williams Bakery, Ethel Corp, BI Chemicals, Biotage, and the University of Virginia. He is now the Senior Director of Business Development for Quotient Sciences in the Philadelphia area. In that position, he supports the drug development community with a variety of research services, including the drug substance development, formulation development, GMP manufacturing, and clinical conduct. Tobi, I will pass it over to you.
Great, wonderful. Thank you, Ashley. And I love that statement. Toby has his own set of rules. I think a lot of people would agree with you on that one. So we will jump in. Um, I do encourage questions. So as we go, we'll have different ideas that we'll encounter. And please speak up. If you think it's something that should wait, please put that in the chat. Or if you want to break in, if you got something really excited, just let me know. All right. So just a little background on me and my role. So really found a lot of value in a book that was interested in defining a person's purpose. And so it encourages everyone you work through the book slowly over time, and you eventually get to the point of like you want to define your purpose in a single sentence, and it's kind of looking at a theme that's really hard to do at the beginning of your career, but it's something that can shape up after you've had a job or two. So I figured that pretty early on, my purpose is to assist scientists developing new drugs. It's just something I really have always felt like something that drives me, that gets me moving. And there's a tremendous amount of highs and lows in pharmaceutical development, whether you're in business development or any other area. But yeah, that's something that, when you get a setback, it's something to pick yourself up and keep going, is that, yeah, we are making progress in something important. So how did I get here? Starting from the bottom and working my way up. Williams Bakery. So my grandfather started Williams Bakery in Mechanicsville. My father bought it from him, and then my brother has bought it from my father. So my brother Carver Williams still runs it to this day. My sister Summer Williams is actually a cake boss over at Shindig. So if anybody's familiar with Shindig's at all or Williams Bakery, we're still plugged into that community. I still go back and help out during Thanksgiving and Christmas rush from time to time. If that looks like a really young starting date, I did start when I was 12. Working a family business. Yep, come on in, ice some donuts. So that's where I got started. Not even knowing that I was going into chemistry. Pretty cool to have some background in baking. I do tend to bake on large scale. My wife gets irritated when I get into the kitchen. It's just everything happens really big. That's just how it's always been with me. When I was studying chemistry at Bluefield College, then moving over to University of Virginia, was fortunate enough to get an internship working as a bench chemist at Ethel Corporation. Learned a tremendous amount there. I would say one of the values of the internship, especially as an undergrad, is working with people in the industry and learning them well enough to get a positive reference. So Jeff Milner, Vasudevan Bala have some really big influence in my early life. Just in that one summer internship have just really helped me refine where I could go. Got a chemistry degree at University of Virginia. Thought I was going to go to medical school. I got told pretty quickly that my academics were not up to snuff as far as getting in. So I had to do a redirect. And it's like, all right, what else do I like? Well, I actually do like the chemistry end. So went to work for a company called BI Chemicals. BI Chemicals is part of Boehringer Ingelheim. They have a massive facility down in Petersburg, Virginia. I understand that's been through a couple of changes of ownership. Julian and Ashley and I were talking about some people common in the chemistry scene, and that was actually Frank Gupton that gave me my first job there at BI Chemicals. So Frank and I have known each other for going back a little ways. Um, Working at BI Chemicals, it was a pretty challenging job working in a plant, working 12-hour shifts, wearing a respirator and moving giant drums of solvent around. Was like, yeah, this is kind of physically intensive. Looked around and had the opportunity to work with Biotage, which was back in Charlottesville. And hey, you get to wear a white lab coat, you get to work 9 to 5. That sounds really cool. So came into Biotage, and Mike Lally was a huge influence there. They were interested in me because I had that industrial background. So even though I was with BI Chemicals for about a year, it was useful to build on that experience because Biotage was looking at removing chromatography out of a lab technique and more towards a manufacturing technique. And so preparative chromatography was the name of the game there. At that time when I was at Biotage, Bob Bickler and a couple of others, Peter Ron, encouraged me to join American Chemical Society. So that's where I've joined and started to attend the national meetings and really enjoyed that community. After going to Biotage for a while, I realized that I wanted to go ahead and get an advanced degree. It does limit you a little bit if you have only a bachelor's and you kind of want to go into more research-oriented stuff. Rather than going into chemistry, I started to look at a master's of science in systems engineering. Systems engineering is kind of the liberal arts of engineering. It's everything that doesn't fit neatly into the other areas. And I did enjoy that program. Unfortunately, I did not get the same kind of commitment from UVA that I wanted. And so I wasn't actually in the program formally. So that means I was taking classes as night school, trying to work on that degree. And that kind of petered out after a while. So it's kind of a misstep there where I wanted to go get a degree and never actually finished it. Now, that doesn't mean that it was wasted time. I did learn a tremendous amount about systems engineering and how to apply that. But it was something that's like, yeah, I think grad school, that next topic that you have coming up is grad school for me. It's not for everyone, and it is a choice that you should go in kind of eyes wide open. Um, Bounced around a little bit while I was in Charlottesville and ended up taking an odd job with the Virginia Department of Health. I was the computer guy for a while, kind of in another unusual role. Learned a tremendous amount about public health and the public health nurses that work there. Tremendous respect for the job that they do there. And that gave me just a lot of empathy for some of the things that people are struggling with that you have really no idea that if you have generally good health and you kind of take it for granted, you'll see some things there that you've never seen before. So yeah, kind of a role is shaping me kind of more as a person from what I learned out of that. While I was looking for my next chemistry job, I did have the ability to interview with two major employers that were really good. Realized that I had worked with a lot of different pharma companies in Virginia, and then started to look outside of it just to see if I could find a role that would better suit me. So, interviewed with a small pharma company called Pfizer, and interviewed with a company called Chiral Technologies. And Pfizer was interested in me for, again, doing large scale preparative chromatography in a plant. And then Chiral Technologies was interested because I had done a good bit of work supporting sales guys. And so it was actually one of the pivot points in my career of like, do I want to continue to go into research or do I want to try commercial side and sales? So again, a decision that I had to talk over and see, okay, did I want to move to Groton, Connecticut? Did I want to move to Westchester, Pennsylvania? And I actually did take the plunge to move into sales with Chiral Technologies. I kind of had an introduction to that because while I was at Biotage, I was a bench chemist. I was spending a lot of time with the sales guys and traveling with them and meeting different clients, different pharma companies. Really enjoyed that lifestyle. Working with lots of different companies means you're working on lots of different projects and things move fast. So doing that with Chiral Technologies full time as a sales guy, that was a lot of fun. Really enjoyed that. Um, Worked with Chiral Technologies for a while, made a change into Variant Instruments selling LCMS systems. That was a harsh lesson because Variant Instruments, very well known for NMR, very well known for GCMS, really not good at LCMS. And that was something that I hadn't done enough due diligence on. So a hard lesson there. I was like, I'm going to stick it out. I'm going to make this work. And then it actually didn't go well. And on my exit interview, HR asked me like, so what do you think it would have taken to make you successful here? I'm like, Albert Einstein couldn't have been successful here. There's too much working against me. And I think you guys really need to reevaluate how you want to work with this instrument going forward. And it wasn't too long after that that they discontinued that instrument line. So yeah, some steps go forward and some steps kind of go a half step back. After Varian, I was really fortunate to land at JRS Pharma. So JRS is Joseph Rettenmeyer and Son. They manufacture excipients. So excipients are everything that goes into a tablet that's not the active ingredient. So the binder, the disintegrant, the lubricant, even in the later days, started working on the colors and coatings, pretty much taught me a tremendous amount about formulation development. And that's a bit of an area that's not as well known, but also very important when you're doing, drug development is the formulation. How do we actually get this fine white powder as a drug substance into a drug product that you can actually consume at the end? Worked briefly for Avantor, another company in the Pennsylvania area. Again, a lot of upheaval into their commercial area. I was really surprised to see Avantor actually ended up merging with VWR, which I think was a good move for them. But in the midst of all of that, was a bit challenging to be there. While I was at Avantor, I had the chance to interview with Quotient Sciences. And I remember pretty clearly the first conversation with the recruiter. They were telling me, yes, they make drug product. I'm like, okay, I understand that contract formulation development. They're like, okay. And he says they actually changed the drug product while the clinical trial is ongoing. I'm like, wait, say that again? Oh, they changed the drug product while the clinical trial is in progress. I'm like, dude, you can't do that. Like that's not allowed. And he said, well, they found a way to do that. Do you want to talk to them more? Do you want to find out how they do that? And I'm like, yeah, I want to find out why they're not in prison. Like that's a big deal. You cannot change that. And it's like, okay, they have found a way to make it work. So I came in and I spoke to him like, wow, this is really lightning in a bottle. They can change the drug product while the clinical trial is ongoing. This is really cool. So again, a little bit of a risk because it's the first time I worked for a company that was headquartered outside of the US. All their operations were in England. They had only 3 employees in the US, and everything that they did was a project that worked at Nottingham, UK. A good bit of difference to work for an international company, and have really actually enjoyed it. It's gone very well. So coming up on my 8-year anniversary with them next week. I think next step, it would make sense to just go through a little bit about what Quotient does. And while we're doing that, will be a bit of an intro into what formulation development, what drug product development actually does. And we will keep going. So this is pretty much the corporate overview for Quotient. We'll move through this pretty quickly. So Quotient Science is a drug development manufacturing accelerator. So you might have heard of Contract Development Manufacturing Organization, or CDMO. We see ourselves as a drug development manufacturing accelerator, and we're just defining that in a new way where we're putting together a couple of different areas of drug development under one roof. And by doing that, we make the actual timeline for the whole project go faster. So integrating services for the development pathway. That's how we do that. So manifesto again matches really well with my personal values of helping scientists develop new drug products, and so it fits very well. One of the things, the hallmarks of Quotient, is that we move quickly, we move fast. So if you look at currency, again, this comes up in an international company. Do you want to pay in euros? Do you want to pay in dollars? Do you want to pay in pounds? International currency is time. Time is going to always come up as a factor. So if you have a project that's going to take 12 months, and then you have an option where the project's going to take 8 months, everybody loves to go faster. It's very rare that we're asked to slow things down. So time is a huge value. If you can find a way to work faster, definitely take advantage of it. If we look at traditional drug development, things are split up into a couple of areas. And this is actually very apparent with big pharma. With big pharma, you may have a company that has evolved with pieces over here, pieces over there. Sometimes you have very, very smart people, but they're focused on just their objective. So you may have people developing drug substance. Somebody in medicinal chemistry has discovered a candidate, and you need to get enough of that drug substance to be able to develop into a product. You have the drug substance guys working on that process R&D. Then you move into the drug product guys. These are the ones actually doing the formulation with the excipients and the tablet compression. And then you have the clinical team who is lining up the human subjects or the patients to get ready to dose that drug product for the first time. In some cases, these people can be in different buildings. They may not see each other. In some cases, in big companies, these may be in different cities. You may have a drug discovery team over here, process R&D in a different place, and then a completely different time zone, somebody else working on drug product, and then a clinical site elsewhere. So it make it difficult for these people to communicate, especially if they're not speaking to each other often. There is a tremendous tendency where someone goes, well, I know that the drug substance is probably going to be ready in December, but I don't want to be rushed. So I'm going to tell them in January. And then the drug product guy goes, well, the drug substance guy was late last time. He's told me January, but I'm not going to really start until February. And so you end up with a lot of dead time. Each of these handoffs can just exaggerate from place to place. So if we start to get these people to communicate and talk to each other better, we can integrate those timelines rather than building in a month of dead time and then another month of allocation on top of that. We can actually say, okay, how is this project going in real time? Do you think you're going to be delivering this week or that week? And so it's possible to integrate the timeline from when drug substance is ready, the QC is complete, the drug product formulation can get started with some non-GMP material. We know when the GMP material is going to be available. Hey, we've got GMP drug product. It's been submitted to the regulatory agency. We know within a couple of days when that's going to be approved. Now we can start recruiting the subjects for the study. So when we combine all those activities, especially with one project manager, we can cut through that silo, improve the communication, and just move the project along much faster. When we look at the different services that Quotient offers on the right side, drug substance, which is process R&D, we see a lot of projects where someone may have made a few hundred milligrams of material and they need to scale up to a few kilos. So we do that kind of work where you look at the actual organic synthesis. I have a friend that uses the term atom economy. How efficient are you in using your different reagents to get to the drug product you need, and then making that GMP. So good manufacturing practices that you have that material qualified to be able to dose into a person. Next step is formulation development, taking that drug substance, that fine white powder, figuring out what the dose is going to be, putting that into the right format. Sometimes that's tablets, could be capsules. Sometimes we get a very poorly soluble drug substance. If it's very lipophilic, very hydrophobic, we may have to do something to that substance in order to get it into the body. So we may use something like spray dried dispersion or hot melt extrusion or micronization. There's a couple of different techniques that you can use, and the different technique will depend on what that drug substance looks like. So that physical chemical properties of the drug substance, because all these different candidates come in, they look different, we have to respond based on how they behave. So that will definitely influence your formulation development. Um, Once you have your formulation in place, then you're actually making the drug product suitable for dosing inside a clinical trial. Now, dosing inside a clinical trial is very different from dosing commercially. Inside a clinical trial is a very controlled environment. Even though these things may have been through significant animal testing, you're not quite sure exactly what's going to happen when you dose it in a healthy volunteer. So we start at very low doses, and then we bring that dose up slowly under a subject where they're actually in residency. They're going to be living in the clinic for several days, and we're going to be watching them very closely, taking vital signs, and we're going to be looking at their plasma to see how is this drug behaving once it gets into the body. So we do that over time. Because we're doing that, we can make the clinical trial material to different standard. Once you've actually gone through phase one, phase two, phase three, and you get a commercial approval from the US FDA or the UK MHRA or whatever health authority is, then you can actually get to the point where you're making large scale commercial batches and you can sell that to any country wherever the doctor prescribes it. Along the way, clinical pharmacology, the phase one study is going to be dosed in healthy volunteers. You have to be extremely healthy to volunteer for these studies. First time I sat down with a physician who was interviewing a potential volunteer for the study, they spent an hour with them. So if you think about how fast most doctor visits are, you might get a physical and spend maybe 10 to 15 minutes with a physician. Every subject coming into a clinical trial is going to spend an hour or so with a study physician, and they go over very specific questions. One time when I came in and sat in on that interview with the doctor was asking, okay, have you broken any bones? I'm like, that's a really specific question. Well, when we do certain types of studies with radiolabeled material, the radiolabeled material will actually go to the site of a fracture in the body and accumulate there. Not really sure what that mechanism is, but it's very consistent. It happens a lot. So if somebody's ever broken a bone, even if it was decades ago, it's something that the physician needs to know, especially if they're going to be participating in a radio-labeled study. Um, the subject said, nope, never had any major broken bones. Okay, and go a little bit further. Have you had any surgeries? How's these habits? How these habits? And then a little bit later, it's like, okay, have you ever been in a car accident? Well, actually, yeah, I did. I was in a car accident when I was in the military. Okay, tell me about that. How did that go? Any concussion? No, no concussion. Did you get any injuries out of that? Well, nothing. I just broke two ribs. And the physician goes, wait, you broke two ribs. Can you show me? Okay, those are broken bones. And she goes back 3 pages in the questionnaire and actually changes that. That's the level of detail that they go into for a healthy volunteer study. So pretty intense questions so that we have a lot of background about each subject before they volunteer, so that once they get into a study and someone says, hey, I was dosed with this drug product and now I'm having a headache. It's like, okay, do you have an idea of how much headaches you have an idea of their or rest of their habits, how hungry they are they prone to anything? Because you've got all that background of a very healthy volunteer, you have an idea of, is this something due to potentially from the drug, or is this something because this particular subject is prone to it? So that side of clinical science is still really new to me, and it's one thing I've learned coming into Quotient, having to appreciate how sophisticated that clinical science is. One of the other things we do is bioanalysis. When you dose that drug product, you want to see how much is actually getting into the blood, how much is getting into the plasma. So you pull samples from the subject every few hours, and you send that out to a lab that runs the LCMS. And that LCMS is actually looking to see that drug substance in the body and see if it's changed, see if it's metabolizing into a different form. Out of that LCMS, you get gobs and gobs of data. So you do need a team to interpret all of that raw data. That's where the data scientist comes in. These guys are just wizards with software packages that are far, far beyond Excel to be able to take the drug product at different doses and see how that's interacting with the human body. And then knitting that all together with drug development consultants. Once we start taking two or more of these services and putting it together into a single program, that's what we call integrated program or translational pharmaceutics. We can do that early with candidate selection. We like to look at salt form A versus salt form B. We can do that in early formulation development where we're looking at a hot melt extrusion versus a spray dry dispersion versus a micronized version. And we can also do it in later development where we're starting to look at what the mass balance is. Once you put a drug product in the body, how does it go? Where does it get absorbed? How does it get excreted from the body? All of that can be very important to putting together a file for the regulatory to get to commercial manufacturing. All right, so the question is, how much time can you save? We actually did a study with Tufts University, and we looked at 20 different case studies of programs that had been done at Quotient. And we looked at them and said, okay, these are what the Quotient timelines were. This is what the Quotient budget was. Let's compare that to if we had done these in a traditional method. And so we provided a lot of data to Tufts University, and Dr. Joseph Damasi with a research assistant, Michael Wilkinson, did a study looking at these 20 different case studies, and they came back and they said, yeah, Quotient is saving, with the integrated program, saving an average of 12 months. I'm like, wow, that's a tremendous amount of time. If you were using separate vendors for all these different activities versus putting them into just Quotient, 12-month savings. When they start to put dollar figures onto that, it comes up to be pretty impressive. It was an average of about $2 million in savings on the programs. So significant reduction in the R&D spend. And then also you're making decisions while you're going through that trial. Because you have the ability to tweak the drug product, dose in a healthy volunteer, look at the bioanalytical, see how much is actually getting into the body, change that drug product, then dose it again, and then see that difference. That gives you better decision making. You're actually doing small changes along the way, which is really helpful. Gives you a better likelihood of success, and then also a simplified supply chain because you're communicating with one project manager and one team. It just cuts down a lot on the handoff from person to person. It just makes it a smoother experience. I like maps. This is a map roughly of how big Quotient is right now. So Philadelphia has GMP, good manufacturing practices, GMP sites for manufacturing solid oral drug product. So tablets and capsules are mostly what we're making. We do have the ability to do some oral liquids, and we do get into some other more exotic formulations such as granulates and powders. Our clinic in the US is in Miami, so that has roughly 144 beds, also has a compounding pharmacy. So we can make drug product at Philadelphia, put it on a plane for a nonstop flight, and then have it ready to dose in Miami within a day or two. So that's a very fast process. If we go over to United Kingdom, that's where the original project started. So Edinburgh is our data sciences team. Any of the clinical data that comes out of Miami or Reading or Nottingham would end up at the Edinburgh data sciences team. Anik is our drug substance development. Nottingham has the ability to do drug product and also a clinic. And then Reading is another GMP facility doing drug product. The very original site is Nottingham, which is really cool. It's actually GMP manufacturing upstairs. They're making the drug product upstairs, and then downstairs is the clinic. So it's actually under one single roof where you can make drug product upstairs, 1 of 6 GMP suites, take it downstairs and put it into one of the clinical wards and dose it there, 1 of 6 clinical wards. It's actually fun. One time we had an auditor come in and they said, can you show me how you actually ship samples? Can you show me how you ship the drug product? It's like, well, we take it from upstairs and we move it to downstairs. It doesn't actually leave the building. It doesn't go through any shipper. It doesn't go through UPS or FedEx or anything like that. It's actually that quick because we can make it, do the release, and then dose it really in a day or so, which is pretty fast. This is kind of the history of Quotient, a number of different companies that we've added over time. Pharmaceutical Profiles, Quotient Clinical, and then more recently moving into Co-Formulate, which is a pediatric specialist, Seaview, which is the Miami site, and then Arcanova, which is now Quotient Anik, which is a new facility where we're doing a lot of the drug substance. So lots of experience. Company's been going on for a while. It's neat to look at the number of molecules. So we look at molecules as one kind of currency for us. The more drug candidates that we touch, the more that we can help with the development. So we're looking at about 150 new candidates every year, about 3,000 candidates that we've seen over the lifetime of the company. So things are moving very well. So that's a quick overview of Quotient. I'll just pause here. Any questions so far? All
I
right.
guess, oh yeah, I do have a quick question. Is there any
Sure.
common, I know sometimes you see commercials on TV selling like like a tablet or so. So is there any name that's where Quotient is under?
Great question. So, Quotient, because we're a contract manufacturer, we don't have any products that we make that are sold by Quotient as a Quotient product. Everything we do is fee for service. So
Okay.
we are making drug products for pharma companies that you would recognize. But because of confidentiality, it's very difficult for me to tell you exactly who we're manufacturing though. I can say that we are making commercial drug product for 8 different products approved in the US. They are very specific needs. So about half of those are for oncology and about half of those are for orphan drugs. So these are not things that you would recognize, but they are out there and they are approved.
Okay, all right, thank you.
Sure. Now, I was happy to support the development of a product that did scale up, and I was. As far as a contract manufacturer, you don't really take credit for that, but it was fun to be part of that. And then the drug product actually ended up getting advertised during the Super Bowl, and I got all kinds of excited. I'm like, look, I was a part of that. That's really cool. So it is fun to see some recognition, to see when something actually does go all the way to commercial. All right, so moving on to a little bit, what is sales? What's the commercial side? I found early on that grad school can be great for certain people, and research, career in research is absolutely fulfilling for certain people. I found that for me, I really like sales, and some people try it and don't enjoy it, and some people try it and really love it. So it's not so much a function of intelligence as much as a function of personality, of whether you like it or not. So um, this is a kind of a brief overview of the sales process, just if anybody's curious about how this works. A lot of this came from Mike Lally, who was a tremendous influence on me and who's still active, not with Biotouch, but with another company in the Charlottesville area. So he said, really, a sales plan is where to go, who to see, and what to say. You know, where do you want to go? What type of company are you trying to work with? Who's the right person inside that organization? And then once you finally connect with that person, what message do you want to give to them? And sometimes you will get a very small window with the right person. So if anyone's familiar with the idea of an elevator pitch, from the time you get on the elevator to the time you get off the elevator, you have to be able to make a compelling presentation of why this person would want to have a second conversation with you. You need to have that in hand. So once you have that message in hand, then you can start doing the prospecting. So identifying the target companies, inside Quotient, and for most of my career, really, it's been pharmaceutical development. I've been focusing on people that have drug candidates that are small molecule organic drug candidates, almost always new chemical entities. These are things that have never been approved before. As far as companies, I work with all different sizes. So small, medium, and big pharma are all part of my region. Big pharma are companies, the top 20, top 25 that you'd recognize. Small companies can be as small as 2 people. You know, 2 guys with an idea and some funding can actually go very far in the development world. Next up is finding the right type of contact. This can change depending on what service or what product you're selling, and also depending on where they are inside that company. So for me, currently, I'm looking to meet people doing formulation development, drug product development, drug substance, specifically process chemists, chemists that are doing drug substance that need to scale up, and then clinical pharmacologists, which is somewhat new to me. This first time at Quotient, first time connecting with Clin Pharm people. As far as the different titles, can be as high as the chief scientific officer. Vice presidents are very common in some of the larger companies. I'm working with the directors and the managers, and those people are usually the ones that get the project started. And then I'll work with all different types of scientists along the way. It's pretty rare for me to work with a chief executive officer, CEO. That did happen this week, so that was kind of fun. But usually it's the CSO, vice president. Those are usually the people I'm talking to. Presenting the right message. Again, this is, I've done a good bit of music. So I grew up playing trumpet at my high school and then played trumpet a bit in the college. And the idea that you rehearse a lot before you actually perform, that you have your message down, that has come up a lot. So having a compelling message ready so that when you do finally get somebody's attention, that you can connect with them and share with them what you think is important based on what you think they might need at the time. So it's really helpful to go and research what a company is doing. I pull up the company's website if they have one, take a look at their pipeline. Even a company that's in stealth mode and may not have a website, usually you can try to find something that's out there or at least some bit of information on what they're working on. If you find that person and you have an idea of what their problem is and you present them a way to solve that problem with the right time at the right person, that's when you have a project. So that's when things can actually get kicking. It is possible to connect with the right person at the wrong time. That's okay. You have a pleasant conversation. You tell them this is what we can do for you when it's right, and then you don't force the conversation. They're trying to solve the problems that are important to them, and then at some point in time in the future, then they'll engage. So it's a matter of just connecting with them at the right point that they need you. Where to meet the right people? Major industry events. American Chemical Society has been tremendous help. American Association of Pharmaceutical Scientists, AAPS, is another big one to meet the formulators. Drug, Chemical, and Allied Trade, which is the DCAT meeting up in New York, and then Chemical Pharmaceutical Intermediate, CPHI, in Philadelphia every year. So these are big major events where I try to meet people that are that I already know, and also try to meet new people. Some of the specialized events, Chem Outsourcing is another group for drug substance. That one's in New Jersey, as well as Contract Pharma. And then the pediatric community, even in the US, scientists tend to go to the European Pediatric Formulation Initiative. So that EUPFI really is the dominant group as far as people doing pediatric drugs, which is becoming a bigger deal for us. Also, meeting the right people, friends of friends. I have this very frequently where smart people will join a company and at some point in time, it just makes sense for them to move on. Sometimes the company's successful, sometimes it's not. Sometimes people are just ready for a change. And so when you have a great experience with somebody and they move somewhere else, it's usually very easy to connect with them. So having that reputation and just being friendly, asking questions, and connecting with people when they're in the new spot, people that I'd never thought I'd see again have popped up in different states at different companies in different roles. It really is a small community. So once you get to know people, it's worth keeping positive relationships. A lot of industry consultants where somebody will end up working with a company for a while and supporting. Sometimes some of the smaller companies don't necessarily need a full-time employee, so they'll hire somebody on as a consultant, and that person will help guide them through drug product development or drug substance. I end up working with a lot of consultants that introduce me to their clients, so that helps. I will just say a word of warning. It's always good to be sincere with networking. LinkedIn has made it very easy to friend and request connections to gobs of people. Really, I try to keep my LinkedIn network kind of close to me. It's good to have that personal connection. So I enjoy that. And LinkedIn's great for keeping up with people with where they are. But yeah, blind requests I found have been a bit challenging. They're not as useful as actually meeting somebody face to face. A little bit about right message, right person, right time. This is partly due to Quotient doing so many different things, so we can interact with people depending on where they are and help them move forward. If somebody's at preclinical, I'm going to ask them about what's their drug substance status. Do they have a process synthesis ready to go? Are they still working at bench scale? Do they need a formulation strategy? And so this is something that's very big. What's your target product profile? What would you like this product to look like when it goes to market? Are you going to be doing a sterile injectable? Are you looking for an oral drug? Would you like your drug to be taken once a day? Almost everybody wants their drug taken once a day, so that modified release can be helpful. Modeling and simulation, first in human strategy, these are other things that we can do for people in the preclinical stage. Once a company has moved into phase one, the conversation changes a little bit. Sometimes we find companies will start with a super basic formulation, just drug and capsule, just straight API into a capsule, and that's all it is. And that's okay to get started, but you're definitely going to want to have a better solution once you get into phase two. So formulation optimization is a big deal. We start asking about their human ADME plan. Human ADME is where you actually put a radiolabel C14 in the drug substance, and then you dose that as a drug product and find out how it goes and moves through the body. So these projects can take a while because you have to line up the radiosynthesis, you have to do the drug product with the radiolabel material, and you have to line up the clinic. So while parts of the program are very simple, putting it all together is quite a thing. So I say it's a bit like taking a freight train to Pittsburgh. Is like it takes a lot of energy to get started. Once it gets started, you don't want to stop it. You want to hit all of those stations along the way and keep rolling along. So human ADME, even though they are seem very simple, it can actually take up to 2 years to get an ADME project to actually go forward. Once you get into later stage, phase two and phase three, asking about commercial manufacturing, we're seeing a lot of pediatric projects popping up. The US FDA has been very forward about requiring companies to develop a pediatric product as either a condition for getting approval or as a requirement after you get commercial approval, you need to develop a pediatric formulation. So we're seeing that pop up with lots and lots of different products. The old solution for pediatrics was take the tablet, grind it up, put it in cherry syrup. This is a pretty harsh way to just throw everything in cherry syrup, and it actually can have some problems. So we have a neat program where we're doing taste optimization with a couple of different flavors based on what the phys chem looks like. And then we also look and say, okay, what's the best formulation? Do we want this to ship out as an oral liquid? Maybe we want it to ship out as a powder, and then we have the pharmacist add water and then shake it and reconstitute it. So that can be a lot of advantages. In some cases, the drug product tastes terrible and you just can't get around that. And so it's like, how do you get a drug into a kid who can be very sensitive to taste that tastes really, really bad? So we have developed a strategy where we can either take a granulate or a very small tablet, mini tablet, coat that with a coating that gives you some taste barrier, and then you can take these small mini tablets or granulates and put them over food like applesauce or yogurt or something like that, and then feed that to the kid. So that's one way to get a very poor, very bad tasting drug into a kid. Those kinds of projects can be challenging. They can take some time, and there's a lot of different ways to explore what's the best, because again, every drug candidate comes through is a little bit different. You're going to have different solutions for different problems. As far as the people involved, I work with different scientists of all different types. So formulation development, product development is the most common one. These guys are most concerned, how am I going to take this drug substance, put it into a drug product? How am I going to make this and scale it up? Medicinal chemists, synthesis process chemists, how these guys are doing the drug substance, spend a lot of time with them. Clinical pharmacology, developing their clinical strategy. How are they going to take this in? Are they going to be going into a lot of healthy volunteers? Are they going to go into a few healthy volunteers and switch quickly over to patients? Where is their clinical site? If their clinical site's going to be in the US, we're going through the US FDA. If it's going to be in the UK, it's going to be in the UK MHRA. And that clinical strategy will shift how you approach your product development. So that's an important conversation to have. I often like to ask people, please bring your clinical scientists in. Like, yeah, but we're talking about drug product. I'm like, I understand, but I really want the clinical scientists to weigh in, because sometimes they'll show up with information that surprises us. So I like to get that communication going. DMPK, biotransformation, these guys are looking at human ADME studies. Quality and regulatory, that can be very important. Again, aligning where we're going to be dosing it with the regulatory strategy. And then also environmental health and safety for highly potent actives. We're seeing a lot of different actives come through that are incredibly potent. And these things can be very useful, but it's also a concern that you don't want to have any of your manufacturing team or scientists exposed to something that could cause unwanted health effects during manufacturing. So the EHS, environmental health and safety for highly potent actives, that they're also important to have involved in the conversation. I mentioned earlier different sizes of companies. Um, Different companies do business with Quotient for different reasons. Big pharma tends to like working with Quotient because we're fast. Sometimes we're faster than their internal resources. Sometimes their internal resources are great and they're just overwhelmed with projects. So we're happy to pick up the overage when they need to move things quicker. Small pharma tends to work with Quotient because we offer so many different services and we can provide all of that as a package. They don't have to go out and qualify multiple different providers. They can get everything they need from us. Medium-sized pharma, there's actually a mid-majors, not quite big enough to be big pharma, but definitely not a biotech. And we adjust because some of these guys have internal resources and they want to use that. And we're happy to move things off the menu that they can do inside. But then we can also fill in the gaps. We can provide those resources that they need. And it's very much a la carte. We adjust based on what they need. We don't feel like we need to tell them, hey, we've got a bioanalytical lab, therefore you have to do all bioanalytical with us. That's not the case. We have a bioanalytical lab. It's great. However, if you have your own lab, we're happy to work with them. So we try to be flexible when we approach those companies. So going to move into a little more science here. Any question about the commercial aspects of the business?
Yes, I believe there are a question by Ashley. She said, regarding finding the right people at industry specific events, do you research people before conference so that you know who to look for, or do you let conversations occur organically?
Yeah, yeah, a little of both. So I love to try to set up meetings with people that I know they're going to be there. So that helps to have something scheduled. But attending the mixers and the receptions and just meeting people, again, friends of friends. If you see somebody you know and then they're talking to somebody you don't know, I've had multiple conversations just open up like that of like, oh, hey, we worked together in the past. And yeah, that organic, being there in person is hugely valuable. So there's a lot of great conversations get started that way. Cool. Brett, did you have
Can I
something?
ask a question?
Yeah, go ahead.
Yeah. So just curious, you talked about trying to find problems that they have that you can help solve. Um, besides just talking to somebody, if you're at an event and you say, hey, like, what are you guys struggling with right now? If you're reaching out to somebody, maybe like cold calling, like you mentioned, like LinkedIn messaging or something
Mhm.
like that, how do you go about finding their problems?
Yeah, great. Asking questions, exactly what you're doing. What kind of problem do you have? So, and then also being responsive to how they answer and kind of exploring that a little bit. So if you hear something, kind of build on that question. um, just being receptive, being a good listener, which is something I tell my kids all the time. Practice being a good listener. Ask the questions, ask an open-ended question, and then see what you learn on that. It is possible to see trends. Like once you start working with different communities, you get an idea of who has these chronic headaches that come up again and again. So finding out who their role is and what type of company they work with, you can have a guess of like, are you struggling with X? Are you struggling with a Y? Have you guys put together a formulation strategy yet? Have you guys put together a plan for your ADME? And I've seen some people just kind of click like a light bulb. They're like, wow, we haven't thought about that. We probably should. Okay, great. Let's talk tomorrow. That kind of thing can be really helpful.
Got it. Thank you.
Sure. All right. So we're going to dive in a little more on the science side. Formulation design space. So this, if anybody's ever done design of experiments, very similar. If you look at what goes into a reaction and the products of that reaction, you have an idea, dependent variables, independent variables. And a formulation, it's very much like that. So you can have certain variables in a formulation that will impact how this drug behaves in the body. Easiest one is the drug dose. The more of a drug substance you put in, 1 milligram, 5 milligram, 20 milligram, then the more of that is actually going to get into the body, with a couple of caveats, but that generally is one of the very first variables that you learn to adjust. You can also adjust a couple of different things that are not that active ingredient, such as a functional excipient. So there are, as I mentioned earlier, some actives that do not like to get into the body. And so they can be very lipophilic, hydrophobic, and you may need to put something in there such as a surfactant or a detergent or something to help actually get that in. So that functional excipient is another one to change. Drug polymer ratio for modified release, what kind of gelling agent you have in there, different coating and composition, all of these things can impact how that drug works once it gets into the body. What we've done inside Quotient is rather than making one drug product, submitting it, and then dosing it, we've actually made a range of drug products. So we'll identify a variable such as dose strength, and we'll identify a second variable such as a surfactant or a functional excipient, and then we'll make drug products at the corner points, a low dose and a high dose, a low amount of excipient and a high amount of excipient, and we'll end up with a design space. So with this design space, we will validate all 4 points of the drug product and submit that to the regulatory agency. Once the regulatory agency reviews that, signs off on it, great. Now we can make a drug product down here starting low dose strength, low amount of excipient. Let's dose that and see how that behaves. Okay, we'll dose that, pull plasma samples, send that to bioanalytical lab, get an idea of how much is circulating. Okay, now let's change the dose strength, move from 1 milligrams to 5 milligrams. How does that respond? Again, same cycle, dose that into healthy volunteers, pull the bioanalytical samples, look at that data and say, okay, now that we have an idea of how dose is working, let's change this amount of excipient. Let's move that up. And so when we move that up at the same dose strength, we're still inside this design space. We can make that drug product, dose that, and then get that bioanalytical data. So within 3 dosing periods, we've got a couple of different data points of what's starting to emerge. Is it more important that we change the dose strength? Is it more important that we change the excipient? And this is a curve that will only come out after you're dosing in healthy volunteers and humans. It's possible to try to do this in animals, but animals have a different digestive system for humans. Even dosing in primates, you're going to get different results than dosing in humans. So by adjusting the drug product while the clinical trial is going on, we can iterate very quickly. And within 3 or 4 or 5 clinical dosing periods, we can escalate and find out what's the ideal inside this whole design space. Where should we be? We may end up at a position that is not expected. So if you have a dose range of 5 milligrams to 50 milligrams, you may end up at 37 milligrams. That's something that the data may lead you there. Out of a percentage of 5% to 25% functional excipient, you may end up at 13% of the functional excipient. Because you've covered this whole space, you're not restricted to any one point on the grid. You can move anywhere inside this design space. And that flexibility is hugely helpful when you're trying to figure out your formulation. This is one of the classical case studies where we wanted to get a drug into the body. The immediate release formulation was this red line here. We had too much drug going into the body, which was over an adverse event. So something like headaches or nausea or something that we didn't want. There was too much drug getting into the body. Over time, we got into the zone that we wanted to be, but that was about 4 hours in. So if you can imagine someone having a headache or having nausea for 4 hours, really unpleasant. They weren't going to like that. We kept the drug product or the drug in the body circulating in that zone for about 12 hours. And then after about 12 hours, we dropped through our threshold. So this pharmacodynamic threshold, we're not seeing the impact on the body that we want. Say it's a cough and cold drug, and we're trying to suppress cough. Well, once you hit this level, the patient starts coughing again. Okay, so it's no longer effective. So with the IR drug, it was causing problems for the first 4 hours. It was effective to about 12 hours, and then after 12 hours, it wasn't actually as effective anymore. So what we did is we added a amount of gelling material polymer called HPMC. The HPMC helps slow down the release of the drug as it broke down in the stomach. And so that allowed the drug substance to release slower into the body. And so we would change the dose, we would change the HPMC, and we did that in dose period 1, dose period 2, dose period 3, and we're tweaking it as we go. And by the time we get to dose period 4, we actually have this green line where the drug came on slower. So we didn't go over the adverse event threshold, but we still were over the efficacy. We're still over the pharmacodynamic threshold. So within the first hour, we're getting drug into the body at the level we want. It raises and then it starts to come back down. But as it drifts down, it stays inside that zone that we want it to, so that by the time we get to 24 hours, you're still having that efficacy. Your cough is still being suppressed. That's what you want. Ideally, you want a drug that's being taken once per day. Occasionally, we'll see drugs that are dosed twice per day, but if you can make something that's dosed once per day, your patient compliance goes way up. So that's where we ended up with the green line. We did have 5 dosing periods here. If we needed to keep tweaking to get a 4th prototype, we could. Because we found success on prototype number 3, we actually did a food effect. So food effect is we had been dosing this on an empty stomach. We got the green line. That's where we want. Now we're going to give the patient a regular size meal, usually a breakfast. And after they've taken a breakfast, then we're going to dose, and then we're going to see, does this green line still match? And sometimes the food effect will push it a little bit. Sometimes the food effect will push it a lot. But that's important for us to determine, hey, are we still getting the performance that we expected? The result of that food effect is going to be information that goes onto the prescribing label of the drug once it's actually approved commercially. Some drugs you will see take with food. Some drugs you'll see do not take with food. Some of these, it can be kind of a minor improvement in performance. Some of them, it can be life and death. There are actually some oncology drugs you absolutely must take on an empty stomach. If you take these drugs with food, can cause a huge overdose. So there are actual very, very real practical reasons to figuring out the food effect, because that can have a huge impact on dosing. A couple of other case studies I'm going to move through here a little bit quickly because I know we're starting to run to the top of the hour. This is a study that we did with Eli Lilly, similar to the last one, but instead of changing the dose, we just changed the polymer, and we knew that we had a preferred region. So this was in the upper GI. If you see these acronyms here down at the bottom, it's the lower in different positions in the GI tract. When we looked at the immediate release drug, we had a good bit of the drug releasing too late. It was actually going past the target region of absorption, and it was just being wasted. It wasn't being brought up into the body. So we started at what we thought was a midpoint, 30% gel. and we found that pushed it too late. The drug was breaking up too late, and so it wasn't in that region of absorption. So, okay, let's do less. We didn't change the dose, we just changed the polymer. We put in 24% gel, and we moved it up, but we still didn't move it up high enough. We're still getting a lot of that drug being released outside of the region of absorption. We dropped it all the way down to 20%, which was actually the lowest level that we had here on our parameter. And we found out that 20% gel put the majority of the drug in that region of absorption. Now we're getting more of the drug in the body. Now we're not having to put on a huge dose in order to get it in. It's actually being very effective. One thing that I think is interesting is that the IR drug up here in the green bar didn't have any gel and was still releasing too late. Here we put some gel, which is normally what slows it down. It actually helped release the drug a little bit earlier and was a more effective drug product. So again, this is one of those things that your model, you may think you understand how it works. Once it actually gets in the human body, it can be a surprise. So in this case, 20% gel actually helped you get better early release, which is what we needed. This is a taste study, so I thought I'd go over this really quick. Formulation A was just the straight active. Almost all drugs taste bad. It's a matter of how bad. So these things are almost always bitter. Sometimes they're sour. Occasionally you can get something that's a little bit salty, but nothing tastes like candy. Usually these things are pretty bitter. So formulation A was a reference, no flavor, no sweetener. Formulation B was a fixed amount of sweetener. So this could be something like 1 mg per mil and no flavor at all. And then formulation C, D, and E were different fruit flavors. So we found that fruit flavors tend to work well with acidic actives. So kind of a sour acidic active with a sour fruit flavor, which is pleasant, helps mask it. So we took these flavors, we gave them to 12 healthy subjects. We did what we call a sip and spit, which is they hold about 20 mils of the drug product in their mouth. They swish it around, they get a lot of taste exposure to it, and then they actually spit it out. So that's very helpful because they can take this, they move it around in their mouth and they spit it out. That means it's not actually being consumed. So after we give them a rest of about an hour, they can actually take another one, swirl that in their mouth and spit that out. And they can rate these things, which is really nice because they can taste more than one drug product in a day. We can actually do about 6 or 7 of these in a day for one person. Out of this, we get a lot of information. We ask them how it smells, how gritty it was, how the aftertaste was. Was it too sweet? Was it too sour? A lot of different ways that we ask them about what their impression of the drug product was. Out of this testing, we found that raspberry was preferred by most of the 12 people. We then said, okay, let's take the sweetener and let's push the sweetener. So then we move from a level 1 to double it to triple it to 4 or 5, and let's find out what the appropriate level sweetener is. It is possible that some of the sweeteners push it too hard. And so if you make something that's saccharine sweet, something that's overly sweet, you can go too far. So finding out the right level of sweetener is just as important as finding out the flavor. Here is one particular product that we did. Is actually very challenging. Sialorrhea is just chronic drooling, and this can be something with kids that have had brain injuries, kids that are experiencing different mental impairments, have a really hard time to just stop drooling. So this is a drug. It was actually off patent. It had been around for a while. The big problem was it was bitter tasting, and when you have kids that have special needs, they're really sensitive to taste, and they could not get them to consume that. So this drug active also had some issues. It had to be below a pH of 5 because if it went over a pH of 5, the drug substance itself would fall apart. It was subject to hydrolysis. We had to find something as a flavor to mask the bitter taste. We had to be careful of certain preservatives. Certain preservatives are not permitted in certain countries, and this was going to be in the EU, so we had to avoid parabens. And then we also had to make this appropriate for kids that were on feeding tubes. So this is something that you actually had to be able to dose in the mouth, but also through a feeding tube, through a nasogastric tube, and not have the drug product fall apart due to exposure to the feeding tube. What we actually found was a really simple formulation that worked really well. 5 excipients, very palatable, no objection from the kids, and which was really important. And then we were able to actually get this commercially approved. So this one actually very much a success. This was with a patient advocacy group, which was a kind of a cross between a private company and a nonprofit, and actually went very well. So we were able to support them with this, and they were able to promote their product, which was really helpful. A couple of other ones, I like this one. This was a drug that was poorly soluble, and we had to put a high level of surfactant in it, and we had to put a high level of alkalizing agent in there to kind of neutralize some of the pH. So this actually worked well, except for one problem. So the sodium bicarbonate, you put sodium bicarbonate in a stomach acid, it's going to start to bubble. That's just normal. You put baking powder and baking soda and vinegar, it's going to bubble. It's going to release the sodium bicarbonate into carbon dioxide. The SLS is very much like a detergent. So that's going to give you a bubble like you blow for a kid's toy. So when you actually gave this to the subjects, yes, it got the drug into the body because the sodium lauryl sulfate was working, sodium bicarbonate was working, but their stomach hurt. You've essentially created a bubble machine inside their stomach and gave them a tremendous amount of discomfort. So we had to take a look at this and say, is there a magic region in here where we can get the drug into the body with just enough SLS, with just enough sodium bicarbonate without making the subject sick? So this was kind of an unusual one that we were trying to get the PK right. We're trying to get the amount of pharmacokinetics, amount of drug in the body to circulate, but also not make the subjects ill. So we use this formulation design space in kind of a different area to minimize an adverse event. So this also was very successful. We were able to pare back the SLS and able to pare back the sodium bicarbonate, make it so that the drug still got into the body, but also didn't cause discomfort. A couple of other ways we can use this, looking at freebase versus different salt forms. There's just a lot of different areas where you can take this. Any questions about the science on how we do formulation development? All right, move on just a little bit to where I think pharmaceutical development community is going. I interact with a lot of different companies inside Quotient, interacting with lots of different teams. It is one of the fun things about my job is that if I can get in touch with a company and have a brainstorm, have people around a round table with a whiteboard and just generate ideas, I really enjoy that. The soft skills of doing this job is a lot of fun, and I think in some cases it's a little bit underrated. There's a tremendous impact of having a technical background, but you can have a technical background and use that in a couple of different ways. So um, I think some of the needs that I see, just from my experience inside Quotient and I'm seeing with clients outside Quotient, project managers. So project managers, it's becoming bigger. In the past, in order to be a project manager, you had to be somebody at like a vice president level. Really, we've got too much work. There's too much that needs to be done, and we just don't have time to wait for people to get to be 30, 40 years into their career to lead a project. So a project manager is someone who has been trained, and there's a couple of different certifications out there where people are really just trying to get different teams to work together to act as that common point of contact, and also keep things on track as far as timeline, keep things on track as far as budget. So you're looking really as a team leader. You're going to end up working with many different personalities. You're going to be working with people in formulation development, analytical development, manufacturing, finance, legal, all these different areas. So strong communication is hugely important. Technical background is very valuable. A lot of project managers inside Quotient have come out of an analytical background, a formulation background, or a clinical background. That being said, actually, one of our really good project managers was formerly a teacher. I was like, dude, you have worked with middle school students. You've kept them entertained. You've kept them on track. You've conveyed complex topics in a simple way. Yeah, that's a great skill set for being a project manager. So, a project manager can come from many different areas, and there's just a huge demand for them. Finding somebody that's a project manager who has experience with pharmaceutical development, that's just a golden combination right now. Other areas of high demand, regulatory experts. If someone is familiar with how the US FDA works, how the United Kingdom MHRA works, helping shape that development strategy. Someone that has the ability to read and understand, to recall facts. It's actually kind of similar skills to somebody who's a lawyer, but it's not confrontational. It's much more of just how are we going to build a plan to meet all the requirements that the government has for us and still be efficient. So regulatory experts very much in high demand. Another one that's kind of unusual, but I think is worth mentioning is environmental health and safety, particularly people that have worked with highly potent actives. Those people are really rare right now, and rare and in demand means that they're valuable. So environmental health and safety with highly potent actives, that's also a very good combination. And then I would also like to say business development. Just a little bit about what I've done. Being close to the transaction between two companies gives you a great opportunity to generate value. I'm working with so many different people in any given week. It is a lot of fun. And when you actually get close to the transaction of a company says, yes, I'm willing to pay you $1 million to do this service, that is it's a great feeling to feel that you can provide that much value. It was actually really funny. One of the first projects I was doing inside Quotient, the client asked us, they're like, can you recommend somebody to do data sciences? It's like, well, actually, we do data sciences. Would you like us to add that to the project? They're like, yeah, that's great. They didn't have to go qualify another provider and added $200,000 to the budget right there just by being open to what they needed. So that has the ability to give just to move the project forward and then also just generate a good bit of cash, which is very helpful for growing the company. Technical background, I couldn't do this job without a chemistry background. Communication, I do emails and video conferences all day, interacting with lots of people. I will say finance has helped. My wife's an accountant. I've taken a couple of accounting classes. Accounting 101, if you can only take one class to move your career forward, Accounting 101 is hugely helpful. Beyond that, I would say Systems Engineering 101. Those are just two classes that are just not chemistry direct, but just have a huge influence on how effective I can be with my day interacting with people. Having a background regulatory, understanding contracting, I get along really well with lawyers. That's very helpful. Knowing that and being literate, I have learned how to interact with folks and move a contract forward, particularly working with people that have different goals and understanding that a person who has this role is going to have different goals from a person who has that role and adjusting to what they need. I do like having to see visible success. I like to see my clients move forward. I like to see their projects move forward. I've been involved with a couple of things that have actually commercially launched and actually helped people. So really happy to see that. Again, I can't ever see my name on a patent. That will never happen. I'll never see my name connected to something that says, yeah, I did that. But I do feel some value in having some, uh, having contributed something to moving those forward. I will say a bit of warning on the business development side. You really need a lot of emotional durability. Sometimes you can do everything right and a project will still fall apart. So there's a high risk leads to high reward, but it's still high risk. I've had two projects this year, very high value projects, clients very excited, ready to go forward. And we had actually signed the contracts for getting things moving. And then we got data that said, yep, the project's not going to go anywhere. It's done. That had nothing to do with my performance, had nothing to do with Quotient not performing, doesn't have anything to do with the client not performing. It's just the data indicated this project was not feasible and it ended. So there is a bit of a letdown. You can't assume that everything you're going to work on is going to go forward. So being able to say, yep, that was awful. Okay, on to the next project. Having that emotional durability and being able to handle, even some stress that you may not be responsible for, you may have to take one for the team. That happens a lot. So just be prepared for that. If you want to get into commercial world, that's going to happen. So I have spent a lot of time talking. I think we've answered a couple of questions, but I have time to continue. If anybody's interested to talk, please speak up.